This is a follow-up lesson to the first lesson on Mendelian genetics, which covered basic monohybrid crosses and the associated vocabulary. This will build on that with a quick review of a monohybrid cross, an explanation and a demo of a dihybrid cross, and then some examples of complex inheritance. So first, a review of simple inheritance with a monohybrid cross. So in this example, it says in humans, free earlobes, which will be shown by F. It's a capital F in this case because it is dominant. It says free earlobes is dominant over attached earlobes, which is represented with the lowercase f because it's recessive. If one parent is homozygous dominant for free earlobes, and the homozygous dominant parent would have a genotype like this, while the other has attached earlobes, since attached is recessive, the only way that a person could have the phenotype attached would be if they had two recessive genes. So we know the genotype in this case is little f, little f. And the question is, can they produce any children with attached earlobes? The Punnett square in this case would look like this. And then you're going to put one parent on each side. So here and here. And then you, of course, bring them down. This is a review from the last lesson into each box. And this is what you have. So in 100% of the cases, 100% of the children from this cross are going to have the genotype big F, little f, or this is a heterozygous, hetero for different, since these are two different alleles. And 100% of them are going to have the phenotype free earlobes. So the answer to the question, can they produce any children with attached earlobes, is no. All right, so that was a review of monohybrid cross. A dihybrid cross is still simple inheritance, but you're looking at two genes or two separate traits at a time. And in this case, in order to do a simple dihybrid cross, these traits have to be on separate chromosomes. If they're on the same chromosome, then that increases the likelihood that the traits will travel together, except for in the case of recombination or crossing over. So it's when you're considering two traits, change from two traits on separate chromosomes. Di for two. So in this example of, well, I'll just say sweet pea, we consider two traits, pea color and pea surface. So you're going to look both at whether or not it's got a rough, or I've also heard it called wrinkly, or smooth exterior, and whether the color is yellow or green. And the question is, what is the predicted phenotype ratio for a cross between two pea plants which are heterozygous at both loci? So if it's heterozygous at both loci, then you know it's going to be the genotype. The genotype is for smooth heterozygous, it's going to be like this, and yellow Now, when doing a dihybrid cross, you have to consider the gametes. You're considering the gametes when you do a monohybrid cross, but you don't really have to think about it because it's just one trait. So the way you do this, the way I've always done it, is I'm going to write it out over here. And now let's think of this in the case of combinations that, we'll just use a sperm because it's a easier one to recognize when you draw. So different things that a sperm could carry. It could be a big S and a big Y. It could be a big S and a little y. That's a little small, I'm not going to be able to write it in there. It could be a little s and a big Y. Or the combination could be a little s and a little y. And so now that's what you end up putting here. Are those different possible combinations that a given gamete could have. Now we know if this was the male over here, then this is going to be the female, but we already know what the combinations are going to be for the eggs because we just did them, but we can draw them anyway. Okay, so now you just are going to do the same thing that we learned last week and that we just did with the monohybrid cross, except for you're going to consider both traits. Now you're still going to write it in this order again, but you'll see that as we go through. All right, so for the trait of surface, we're going to have big S, and then big Y, big Y. I'll keep going down this way. So we're going to have big S, big Y, big S, little s, big Y, big S, little s, big Y, little y. And then next column, big S, big Y, little y, big S, little y. Anyway, you get the idea. So I'll just continue to work through these. Oh. All right, so now we've got our large dihybrid cross Punnett square done and the predicted phenotype ratio. So let's see what phenotypes we have here. I'm going to switch to a different color. So let's look first for the ones that are smooth and yellow. Now remember, smooth and yellow are the dominant. So I see smooth and yellow here, 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 here. Okay, so I've got 9 that are smooth and yellow. Now let's look for smooth and green. So I've got 3 that are smooth and green, smooth and yellow. Oh, I'm sorry. Yellow and rough. So 3 that are yellow and rough. And then I've got one last one. That is rough and green. Okay, so the ratio is 9 to 3 to 3 to 1. And that is how you do a dihybrid cross. All right, so even a dihybrid cross is an example of simple Mendelian genetics because it follows the law of dominance, meaning that you're looking at a simple relationship between a dominant and a recessive allele, where in the situation of a heterozygote, one is going to hide or overshadow the other, and the phenotype for a homozygous dominant and a heterozygote is going to be the same. But that is actually not the case with all traits and all genes and alleles. So here are some examples of what we consider to be beyond simple Mendelian genetics, and we'll look at each of these in detail as we move through, but here's a brief introduction. Sex-linked traits are those that are on the sex chromosomes. So those are the chromosomes X and Y. Females, of course, have 2 X chromosomes. Males have an X and a Y chromosome. So that's the only chromosomal difference between males and females. So you already know that there's quite a bit going on there. But there are other alleles and genes not associated with sex determination that are on those chromosomes as well. Co-dominance, this is a situation where in a heterozygote, you have both traits. With multiple alleles, this is something like in the example of blood type, where instead of just, and we just had the one with the free and the attached earlobes, it was either or, two possible alleles. In the case of multiple alleles, you've got more than two. So for example, with blood type, you can have an A allele, a B allele, or an O allele. So you've got greater than two allele possibilities. Incomplete dominance. Now with codominance, you've got both traits showing in a heterozygote. In incomplete dominance, the heterozygote is going to be showing a trait that's midway between the two allele possibilities. And again, I'll go into detail with that. Polygenic, these are things that are determined, traits that are determined by multiple genes. So for example, height, eye color is not just a single gene. Body type, I mean, there's a lot of these that are polygenic. All right, we'll get started with sex-linked traits. And the sex-linked trait that's an example here is having hair in the ears. Only males have hair in the ears. Now remember, females are XX and males are XY. Only males show the trait of hair in the ears because the hair in the ears gene is on the Y chromosome. That also means that if you're a guy and your dad has hair in his ears or on his ears, you're going to have hair on your ears because that's the only possible Y chromosome that can be in the mix. There's other examples of sex-linked traits. Color blindness is on the X chromosome. Muscular dystrophy, hemophilia, those are two disorders that are carried on the X chromosome. Now, most X-linked traits like these, color blindness is the one that we're most familiar with, affect males because they have only one X. So the reason for that is color blindness is recessive in females, or it's a recessive trait. So if it's here, we'll say that's for color blindness. And then just the regular X is non-color blind. A male only has to get one to be colorblind because there's no other X. But a female like this is going to be non-colorblind because the non-colorblind X is dominant in this situation. So that's why you have a higher number of colorblind males than colorblind females. Oops. So let's dig into that a little bit deeper. This just is a test for color blindness. I don't know if it'll work on your screen or not, but if most people are not colorblind, so this kind of gives you an idea of what a colorblind person sees. So most of us can make out the shape of this 2 here. But as you can see, in the case of a colorblind person, they can't. And if you are colorblind, then until I trace the outline of that 2, these 2 looked the same. All right, let's work through some of these sex-linked trait questions. Let's see if I left myself space. I did not. So I will have to kind of zoom in, I think, into some little spaces to do this. Okay, can 2 colorblind parents have a normal son? Well, let's think about that. For the male to be colorblind, we know that his genotype, if dad's colorblind, his genotype is this. If mom is colorblind, what does her genotype have to be? Well, it would have to be this because otherwise it wouldn't show. So is it possible for them to have a normal son? No, because there's not a single X chromosome in the mix here between these 2 parents that isn't colorblind. So they couldn't have a normal son. They couldn't have a normal daughter either. Oh, that's the next question. No, because there's no, even in the case of a female, it's always going to be, regardless of which one of these, dad can only give this one to the female, and regardless of which one, each one of these it is, you're still going to be in this situation. Can 2 normal parents have a colorblind child? Well, for a male to be normal, he would have to have this genotype. For a female to be normal, she could have this genotype or this genotype. So could these two normal parents have a colorblind child? Well, that's going to be a normal female, normal male. That's going to be a normal female. She's going to be a carrier, but she's not going to have the trait because the non-colorblind is dominant. But this son is going to be colorblind. So can two normal parents have a colorblind child? Yes. And what's required is the mother would have to be recessive or a carrier. Can a normal daughter have a colorblind parent? Well, we'd have to look at this situation. Or and in either of these cases, you could have a normal daughter. In this case, you're definitely going to have a normal daughter because if dad's colorblind and mom is not, then the X chromosome from mom is always going to overshadow the colorblind chromosome from dad and not show the phenotype, just be a carrier. And in this case, if dad's normal, it would be the same thing. The X chromosome from dad would be dominant to the X chromosome from mom. So that answer is definitely yes. Can a colorblind daughter have a normal parent? Okay, so in that case, her genotype would be this. Now, dad has to be colorblind because she didn't get a Y chromosome from him. Mom could either be colorblind or a carrier. So the answer is yes, but only in this instance, that mom is a carrier. Okay, hopefully that helps you understand that. It's good to think about those and work with those questions and things in that way. You could have done more Punnett squares if you needed to. I did, I think, one. And then you can also I almost always, at the very least, write out the genotypes just to visualize and get an idea of what I'm working with. All right, so we'll leave the sex-linked traits for now and look at incomplete dominance. So in the case of incomplete dominance, you have no dominant allele. And the two alleles like mix, or you end up, like I said, with something sort of midway between the two. So in this instance, it's a mix of color where it's either red or white. Now what you're seeing here, let me switch my color. This designation is because we don't use a lowercase letter because there's not a situation of recessiveness here. So the little apostrophe is used in this instance. So if a homozygous red is mixed with a homozygous white, the heterozygous genotype is midway between the two, a pink. Another example of this could be if you had straight hair and curly hair, and then the heterozygote was like a wavy hair, which can be the case in the coats of some mammals. That's another example of incomplete dominance. I think we have some practice with this in the lab. Codominance is also an instance where one is not dominant over the other, but it's not a midway, but rather you're going to see both traits fully. So in this instance, this represents the allele for brown, and this is the allele for white. This horse may appear to be a combination of the two, but if you were to actually like pluck a hair from anywhere on this horse, the hair is going to either be white or brown. So both alleles are being expressed fully. It may appear to be a combination of the two, but it's both being expressed fully. So that's codominance. Another example of codominance is blood type, where if you have an A allele and a B allele, then the blood type is AB. And we talked about this in class, So you've got your A proteins, or I think I refer to them as name tags, and your B. Well, here's the codominants. So you see, you're still seeing each of those fully expressed. It's not a midway, it's both of them showing up. And so that's codominance. And it's easy to get confused with the difference between codominance and incomplete dominance. But for me, I think of the incomplete dominance as a midway point. They're sort of meeting in the middle, where codominance, they're both showing up fully. Blood type is also an example of multiple alleles. So you've got the codominant example that we just looked at with A and B. And then you've also got O. So O is recessive to A or to B. But you've got 3 allele possibilities here. And that's what makes it a situation of multiple alleles. And the way that that is determined, let me see what I can't remember what I have on the next slide. Yes. So the way that that is shown, this is the allele for O. This is the allele for A. And this is the allele for B. So if the genotype is one allele for A and one allele for O, you're going to have blood type A. If it is A and A, you're going to have blood type A. So here's your phenotype. And then it says genotype. And then this is just showing who it can receive blood from. And we talked about that in class as well. And the reason for that, let me go back to the other slide. One more. The reason for that is if you have blood type B and you were to receive A, your body's going to recognize these little round name tags as non-self and freak out and have an immune response. O doesn't have a circle or this little diamond doohickey on there. So blood type A, blood type B, or blood type AB can all receive O blood. O can't receive AB, B, or A because it doesn't recognize these. If the genes are a combination of the allele for O and the allele for B, you're going to have B, the same with BB. In order to get the AB blood type, that's the codominant example. So you're going to have one allele for A and one allele for B. And to get O, it has to be both for O because it is recessive. And here's a practice with this. So in this case, the phenotype here is type A. And this individual would be type B. Let's work the Punnett square. So in this case, 25% chance it would be blood type AB, 25% chance child would have blood type B, 25% chance blood type A, and a 25% chance blood type O. But that's because it was a combination. If one of these had been type A by way of this or type B by way of this, then it would have changed it. All right, let's do this other practice. So which blood type are you if you have 2 A alleles or 1 A allele and an O allele? You are blood type A. 2 B alleles or a B allele and an O allele, blood type B. This is type AB and this is type O. Remember again, this is the codominance. And this is homozygous recessive. All right, polygenic traits. The easiest way to determine if something is polygenic is whether or not it has a range of possibilities. Oftentimes the range will be in like a bell curve. For example, height you've got some really tall people over here, some really short people over here, and most of us fall somewhere in the middle. Same with a different skin pigmentation, hair, body type, body size, muscle mass. And what that means is that there's multiple genes on different chromosomes that are all influencing the determination of these polygenic traits. All right, that's it. The lab for this week is going to follow right behind this lesson, and I will work through all of those problems in the lab one by one. So it's going to really tie in with this and help you prepare for your exam, where you will have to really understand these different types of inheritance and also be able to work the Punnett squares in applicable instances.